Hey guys, my name is Tim Merton. My friends call me Nitro. I've been studying for the MCAT for a while and will be testing here in a few weeks, but I thought I'd share with you some of the information I learned about amino acids, what's important, what's not important for the MCAT, the most high yield stuff to know. To begin with, why are amino acids so important for the MCAT? They're actually the highest yield topic on the MCAT. Essentially, it comes down to how many subjects can amino acids be tested on within the MCAT. For biology, they are the building blocks of proteins and muscles. In biochem, they are the monomers of peptides of proteins that can be reacted to form intermediates to the Krebs cycle, like alpha-ketoglutarate and oxaloacetate. In gen chem, they can be tested in the form of zwitterions and the equal and opposite charge within the backbones. In OChem, they are organic molecules composed of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. And in psychology, they can be tested as precursors to neurotransmitters and other neuroactive molecules. So that's 5 out of 8 subjects on the MCAT that amino acids can be tested on. It's worth knowing. While we're zoomed out at the big picture, we can talk about the central dogma of molecular biology, DNA to RNA to proteins. We'll start by looking at our cell here. Within our nucleus, we have our chromosome made up of 2 sister chromatids. Each chromatid is composed of chromatin fiber. As we zoom in further, you can see the chromatin fiber is bound in these units called nucleosomes. Each nucleosome is composed of individual histones. And this bound together form of chromatin is called heterochromatin. So it's the inactive, silent, densely packed methylated form that cannot be transcribed. As we unwind those, the histones become acetylated. It loosens their packing and allows sections of DNA to be available for transcription. As we zoom out further, we now have our strand of DNA. DNA can be duplicated through DNA replication. Then we have the process of that transitions from DNA to pre-mRNA. Remember, as we transition from DNA to RNA, we have sections of the sequence that are non-encoding. These are called our introns. You can see a couple here. These introns will be excised during the processing of the pre-mRNA, while the exons, the coding sequences of genes, will transition as mature mRNA. In this processing, before we leave the nucleus, we remove the introns, as mentioned. We attach a 5' cap, and we also attach a 3' poly A tail. As our now mature mRNA transitions from the nucleus into the cytoplasm, it will be bound by a ribosome in the process to begin translation. The ribosome attaches at the 5' end of mRNA and transitions to the 3' end. So remember, it runs 5' to 3', reads it from 5' to 3'. The first section of the ribosome that the mRNA will be bound is the A site, so that's your amino acyl site. This is where a charged tRNA, so remember transfer RNA, it's charged because it's attached to an amino acid. In this case, the amino acid is proline, PRO. But how did the tRNA know to go grab proline? That's due to the anticodon here. The anticodon is what it sounds like, the inverse of the codon for proline. So down here, within the mRNA sequence, you see that we have the codon for proline, which is our GCC. The inverse of that, accordingly, would be the anticodon on the tRNA, CGG. So the tRNA attaches into the ribosome, attaches onto the mRNA, and the ribosome keeps chugging along from 5' to 3'. As we transition, we go to the P site, which is our peptidyl transferase site. This is where all the magic happens. This is where the peptide bond between amino acids in the formation of a peptide chain occurs, is within this middle P site of the ribosome. It is enzymatically catalyzed. After attaching these amino acids through the peptide bond, the ribosome keeps moving and the tRNA enters the E site. E for exit. So this is where the tRNA is let loose back to go back into the cytoplasm to go pick up, in this case, this CAG was the anticodon for L, for leucine, which the codon for that was GUC. So the anticodon on the tRNA is CAG. This form of tRNA is called uncharged because it's not carrying around its amino acid. Where did the tRNA get the amino acid, you may ask? The amino acids, there's two ways that there can be the amino acids can be in the cytoplasm. That is through biosynthesis. Our body can make them de novo, can make new forms of these non-essential amino acids. Part of the list of the amino acids are non-essential for our diet, so we don't have to ingest these amino acids because we can make them. Our body can make those. The amino acids that are essential for us to consume in our diet because our body cannot make those amino acids. It's not important for the MCAT to know the difference between which ones, like the list of amino acids that are essential and non-essential. That's not within the scope of the MCAT. You should know about it and know that it can be tested in the form of a passage, but if they're going to test you on that subject, they'll tell you whether it's an essential or non-essential amino acid. I'll end this video with the list of what I think is most important to know about the list of 20 amino acids. First of all, you need to know the names of the 20 amino acids that are encoded by our genome. Our DNA contains the codons to code for these 20 proteogenic amino acids. There are a few extra ones that are biologically relevant in very specific cases that we don't need to worry about. Things like selenocysteine that might be listed on some internet sources that you see. That's not generally tested on the MCAT, so don't worry about that one. It's very specific. But do know the names of the top 20 proteinogenic amino acids. The 3-letter abbreviations and the 1-letter abbreviations. So knowing that proline is PRO and that its 1-letter abbreviation is P. And remember that there's a few strange ones in there, like asparagine is N and glutamic acid is E. There's a few strange ones, so keep an eye out for those and try and figure out a way to remember those. 4 is structures. I have divided between the fundamental backbone of amino acids and the unique R group side chain. So the generic backbone is the amino group attached to the alpha carbon attached to the carboxylic acid. Each amino acid is special in that it has its unique R group, right? That's what gives it its unique characters, and the rest of these things to know are relevant to the R group, right? So the physiochemical and functional classifications, there are 3 basic amino acids. There are 2 acidic amino acids. Know the difference between hydrophobic and hydrophilic, aliphatic versus aromatic, charged versus uncharged. So know the functional and physiochemical classifications for all of your amino acids. The net charge at physiological pH for those chemically charged amino acids. Unique features. Know that, for instance, glycine is the only achiral amino acid because it just has a hydrogen on its side as its R group, as its side chain. Also know that glycine is considered an alpha helix breaker because it's so flexible that it can mess with these secondary structures. Know that cysteine has its sulfur within its R group that can bind with another cysteine to form those disulfide bonds. Number 8 here, I listed side chain pKrs. That is the pH at which the R group loses its hydrogen in a titration curve, and I'll cover that in a later video. Know those side chain pKrs for the electrically charged amino acids and cysteine. So I think there's 7 altogether. 6 or 7. pKr, yeah, like I said, is the pKa for the functional groups of the side chains of the amino acids. And then lastly, knowing the start and stop codons. So there are 3 stop codons and 1 start codon. You can find different mnemonics to remember those online. All right, I will leave you with that list of, I think it's a pretty good place to start, and I'll start diving in a little bit deeper into amino acids as we move forward. I'll see you in the next one. Thanks.